In this video, we're going to take a look at meiosis. Meiosis can be broken up into two cycles, meiosis I and then meiosis II. And they're essentially a copy of each other. We're going to go through PMAT twice. PMAT being prophase, metaphase, anaphase, telophase, and then finally cytokinesis at the end. So we're going to describe in this video how a cell, a 2N diploid cell, becomes 4 haploid cells. So let's go ahead and dive in. Now, before we actually get into PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis, before we jump into that, we need to first discuss something that happens in the S phase where DNA is replicated. This is important because the DNA is going to get replicated and it's going to get doubled. And at this point, now meiosis I is ready to begin. The chromatids will join at the centromere, and you'll notice they kind of take like an X shape, and they join at the center. Now in prophase I, chromatids condense into chromosomes, and we're going to highlight a few things that happen in prophase I. Number one, the nuclear membrane will dissolve. You probably didn't notice that, so let me back up. Now look at the nuclear membrane, which is the very distinct black border of the nucleus. It's going to start to break up. It's going to disintegrate, dissolve. It's going to expose the DNA now in the cell. So that's thing number one. Now in prophase I, again, we have three more other things that are going to happen, and that's the tetrad is going to be formed. The chromosomes are going to align, the homologous chromosomes, mom's chromosomes and dad's. Moms can be the reds and dads are the greens. Or at the bottom you have another set of chromosomes. Remember that there's 23 total pairs of chromosomes that are going to pair up, and these are homologous. So the tetrad were formed, and here I have them numbered 1, 2, 3, 4, so you can see them. And something else interesting that's going to happen is that the genes are going to cross over. You will have certain sections of the genome in the chromosomes that will actually cross over and bind to the other side. They will mix and match. If you remember in class, we discussed the analogy of two students wearing two very unique outfits, and if they start switching socks from one foot to the other, or shoes, or shoelaces, or glasses, or hats, sleeves, buttons, and if you make this new person with a combination of those two outfits, you end up with something somebody very unique. So you have two unique individuals making an even more unique individual, in this case, with the genes being crossed over. And then lastly, something else that's very important is the spindle fibers, those gold rods, if you will, those strings. Now, anytime that you see the chromosomes being aligned and moving around the cell, it's really important to consider that they're not moving around by themselves. They are actually being moved by these spindle fibers. They're little nanorobots that attach to the centromere in each chromosome, and they actually usher these chromosomes to the very specific places they need to go. So, having said that, we move on to metaphase I. So, as I mentioned before, the spindle fibers are the ones that are moving the tetrads into position, and they move them in a very specific place called the metaphyseal plate, the equatorial line, the middle of the cell. It is equidistant basically to the poles of the cell. And once they meet at that metaphyseal plate, what's going to happen now is anaphase. Think ana A. They're moving away from the middle and they're going to the poles. And again, notice how these chromosomes are unique to each other. After anaphase I, we go into telophase I, where the nuclear envelope, if you notice, will start to redevelop. The nucleus is coming back. The nucleus is coming back and it's making a whole new cell, and this, what once was one cell, is now going to become two cells in cytokinesis. Two cells with haploid information. Now we jump into this second phase, meiosis number 2, the second division. Now in prophase, so again, we're starting all over again. Let me back up. We just finished PMAT 1, meiosis 1, and now we're moving into meiosis 2. Notice that there's a sub 2 in each one of these. So, but it's the same thing as before. So in this case, now it's prophase 2, where the nuclear envelope disintegrates again, and the new spindle fibers are going to form. Now, I'm not going to show them in this round, but just remember that anytime that these chromosomes move, it is because of the spindle fibers. So they're going to get moved to the equatorial line, right, equidistant to the poles, to the middle, And then right after metaphase 2, we have anaphase 2, where the centromeres, the sister chromatids, are now going to migrate opposite to the cell, each other in the cell, and that is anaphase. And then we're going to have telophase 2. Telophase 2, the new nuclear envelopes are now forming again around the chromosomes. And at this point, they actually start to uncoil and they take less of an organized nature in this case because now the DNA can kind of relax because now it's being protected again by its nucleus. And then finally, oh, let me back up actually. Let me back up. So notice again with the cell, it's starting to actually break up. You have what's called a cleavage furrow, and what's happening is that the ends of the cell are starting to kind of pinch in the middle. And eventually, as they pinch in the middle, they're going to meet and they're going to completely divide the cell into now 4 distinct cells. And when I say distinct, I mean distinct. If you notice the genes, the chromosomes in each one of these nuclear membranes, they are completely unique to each other. None of them are alike. None of them are the same. And the reason for that is because of what happened earlier in prophase where the genes, the chromosomes crossed over, where certain sections switched spots. So making 4 distinct, unique genetic haploid cells. Okay, well, that does it. That does it for meiosis 1 and meiosis 2. I hope this helped, and good luck in your study.